In the previous video tutorial, we had seen that kidneys regulate pH by two fundamental mechanisms. The first one is reabsorption of filtered bicarbonate, and the second mechanism is excretion of H+ and formation of new bicarbonate. Right? So in this video tutorial, I am going to discuss about this first mechanism, that is reabsorption of filtered bicarbonate. Now to understand this mechanism, suppose this is the renal tubular epithelial cell. This is the renal tubular epithelial cell. On this side, we have a lumen, that is tubular lumen, and on this side, we have blood. One more thing is that whenever the cell membrane, it faces towards the lumen, we call that cell membrane as a apical cell membrane. Whereas the remaining cell membrane, that this part of the cell membrane, we call it as a basolateral cell membrane. Right? In the previous video, I had also told that both of these mechanisms, they are carried out by the kidneys by the process of H+ secretion. Right? So, to carry out this mechanism, that reabsorption of filtered bicarbonate, the first step is H+ secretion. So, first H+ secretion will occur, then it will lead to certain sequence of events which ultimately results into reabsorption of filtered bicarbonate. Right? So, the first step is the H+ secretion, which is H+ transported from the cell to this lumen. Now, this transport is carried out by the specific transporter which are present on apical cell membrane. Now, inside this lumen, there is a presence of filtered bicarbonate. It immediately binds with this H+, this filtered bicarbonate immediately binds with the H+ and forms the carbonic acid, that is H2CO3. Now, on this apical cell membrane, one enzyme is present. It is called as carbonic anhydrase 4. This carbonic anhydrase 4, it causes the dehydration of this carbonic acid into water and carbon dioxide. Right? As soon as this water and carbon dioxide are formed, they are transported back inside the cell. So, we have H2O and carbon dioxide inside the cell. Remember, carbon dioxide is the highly diffusible molecule. Almost all the barriers in our body are crossed by this carbon dioxide by the simple diffusion. Right? Whereas for the water, there is a presence of aquaporin channel. So, both of them are transported inside the cell. Now, inside the cytoplasm of this cell, there is one more enzyme present which is called as carbonic anhydrase 2. See, here we have also carbonic anhydrase, here also we have carbonic anhydrase, but both are isoenzymes. So, this is the fourth isoenzyme which is present on the apical cell membrane. This is the second isoenzyme which is present in the cytoplasm. So, here this carbonic anhydrase, it will form carbonic acid, that is H2CO3. Now, you might be wondering that here carbonic anhydrase is converting carbonic acid to the water and carbon dioxide, whereas inside the cytoplasm, it is exactly doing reverse. Why it is so? So, for that, see, here there is a continuous influx of H+ and HCO3 minus, right, and continuous removal of H2 and CO2. So, because of this reason, this forward reaction is favored. Whereas here, what is happening? Continuous influx of H2 and CO2, and there is a continuous efflux of H2CO3, that efflux that we will see in the moment, right? So, because of that, this carbonic anhydrase is favoring reverse than this reaction, right? Now, this H2CO3 immediately gets dissociated into H+ and bicarbonate, that is HCO3 minus. This H+, it can be again secreted inside the tubular lumen. Whereas for this bicarbonate, it is transported here in the blood. And for this transport, specific transporters are available or present on this basolateral side of the cell membrane. So, this is the overall simplified process of reabsorption of filtered bicarbonate. Right? Now, along the different parts of the nephron, the capacity to reabsorb this filtered bicarbonate is also different. And here I want to highlight the 4 different parts of our nephron. One is proximal tubule, second is thick ascending limb of loop of Henle, the third one is distal tubule, and the fourth one is collecting duct. In the proximal tubule, approximately 80% of all the filtered bicarbonate is getting reabsorbed. So this is the major portion of bicarbonate is getting reabsorbed in this proximal tubule. In the thick ascending limb of loop of Henle, approximately 10% of the filtered bicarbonate is getting reabsorbed. In the distal tubule, it is 6%, and in the collecting duct, it is approximately 4%. Right? Now, this is the important difference in the different part of the nephron. But different part of this nephron, they also employ different H plus secretion mechanism as well as different bicarbonate exit mechanism. So let's first look at this different H plus secretion mechanism. So H plus secretion is different in the different parts of the nephron. What is the difference, right? So in the proximal tubule, for the H plus secretion, there are two different types of channel. One is NHE3, that is sodium hydrogen exchanger 3, and H plus ATPase. This H plus ATPase is the V type. In case of thick ascending limb of loop of Henle, it is the both transporters as we had seen in the proximal tubule, that is NHE3 and H plus ATPase. These both same transporters are available in this thick ascending limb of loop of Henle. In the distal tubule, actually there are two parts. One is early distal tubule and second one is late distal tubule. In case of early distal tubule, the transporter for H plus secretion is sodium hydrogen exchanger 2, and in case of late distal tubule as well as in case of collecting duct, both employ same mechanism for the H plus secretion. And here two different transporters are there. One is H plus ATPase, which is of V type, and H plus K plus ATPase. H plus K plus ATPase. Now this H plus K plus ATPase, it is of the antiporter. Antiporter, that means potassium is exchanged with the hydrogen atom. Now we had seen the different mechanism for H plus secretion. Now let's look at the how different parts of the nephron employ different mechanism for this bicarbonate exit from the cell to this blood side, right? So bicarbonate exit. Now in case of proximal tubule, there is a sodium bicarbonate co-transporter 1 is present in the proximal tubule. In case of thick ascending limb of loop of Henle, we have the same transporter, that is sodium bicarbonate co-transporter 1. Along with that, we have two more transporter. One is anion exchanger 2 and chloride channels. Remember, chloride channels, they are meant for chloride transport, but sometimes bicarbonate can also pass through this chloride channel. In case of distal tubule, here also there are two parts, that is part of early distal tubule and late distal tubule. Early distal tubule employ anion exchanger 2 for this bicarbonate exit, and the late part of distal tubule and collecting duct both employ anion exchanger 1 for the bicarbonate exit. Now let's take a brief look on all these different transporter. See, this NHE3, this NHE2, so this NHE, that stands for sodium hydrogen exchanger, right? Here what this transporter does is, it moves sodium inside the cell and hydrogen outside of the cell towards the lumen. So both are exchanging. Now this transporter, it is of the secondary active transport. Why? Because it is utilizing the sodium gradient which is created by sodium potassium pump. So this is the secondary active transport. About this transporter, that is H plus ATPase V type, which is also present over here, H plus ATPase V type. What this transporter does, that it is a primary active transport and it simply secretes H plus into the tubular lumen by utilizing ATP. The third different transporter is H plus K plus ATPase, which is of the antiporter variety. That means H plus is secreted inside the tubular lumen in exchange of potassium being absorbed from this tubular lumen to the cell, right? So this was a brief discussion about the different transporter for H plus secretion. Now let's look at the bicarbonate exit. This NBC, it stands for sodium bicarbonate cotransport. It is present on the basolateral side of cell membrane, and it transports bicarbonate from the cell towards the blood along with the sodium, right? If we talk about this anion exchanger, it is over here and over here. Anion exchanger, it exchanges anion. The first anion which is transported from this cell to the blood is the bicarbonate, and this is being exchanged with the chloride. So that is about the anion exchanger transporter. And this chloride channel, it is very simple that these channels are primarily meant for chloride transport, but bicarbonate can also utilize this transport to get exited from this tubular epithelial cell. So this was all about reabsorption of filtered bicarbonate. In the next video tutorial, I will discuss about excretion of H+ and formation of new bicarbonate. If you have any query or confusion, please write it down in the comment section below. Thank you.